Roberts, and today's date is September 10th, 2021. Muscle glycogen is used during high-intensity physical activity, which is considered to be anaerobic exercise. Anaerobic exercise is exercise that is fueled by energy produced without oxygen. During high-intensity exercise, muscle glycogen undergoes glycogenolysis to create glucose, which is used during cellular respiration to produce ATP or energy. This process is important because muscle glycogen provides an instant fuel source, which allows for strenuous activity. Muscle glycogen is also important in high-stress situations, such as the flight or fight response. Glycogenolysis breaks glycogen down into glucose 1-phosphate to be used for glycolysis. This process is done as glycogen phosphorylase removes glucose 1-phosphate from the glycogen structure by breaking glycosidic bonds. A deficiency in glycogen phosphorylase enzyme would mean that there is not a sufficient number of enzymes for glycogen metabolism. Glucose 1-phosphate would not be formed and converted into ATP, which would affect the production of energy. Although high-intensity physical activity would be affected, long walks would still be possible despite this deficiency. Anaerobic and aerobic are two types of exercise. As mentioned earlier, anaerobic exercise consists of high-intensity exercise such as sprints or weightlifting because they require a fast, almost immediate energy source that is created without oxygen. However, aerobic exercise is physical activity that is low or of moderate intensity. A walk is an example of a low-intensity exercise, and this type of exercise does not require an instant fuel source. For this reason, glucose that is consumed through diet is used in cellular respiration to produce energy. As a result, the pathway of energy production is not altered or remains unaffected with a glycogen phosphorylase deficiency. Glycogenolysis is a process mentioned in which glycogen is transformed into glucose to be used for energy production. Glycogenolysis can occur in the liver or in the skeletal muscles. As mentioned before, muscle glycogen is used to produce fuel during physical exercise. However, liver glycogen is used to maintain blood glucose levels during the fasted state. For this reason, eating at night is not required to maintain adequate blood glucose levels because it is maintained by the liver. Although both pathways use glycogen phosphorylase, a deficiency in muscle glycogen phosphorylase would only affect glycogen metabolism within the muscles. Furthermore, muscle cells lack glucose 6-phosphatase, which allows for the release of glucose from cells. Because of this, it is impossible for muscle glycogen to maintain blood glucose, which further demonstrates that a deficiency in muscle glycogen phosphorylase would not require the consumption of food at night to maintain adequate blood glucose levels. There is no need to be concerned for excessive lactic acid accumulation because of the glycogen phosphorylase deficiency. Because of this deficiency, muscle glycogenolysis is hindered, which prevents the production of glucose 1-phosphate and prevents anaerobic respiration, which would inhibit the production of lactic acid, because muscle glycogen creates an energy source to be used in anaerobic respiration. For this reason, excessive lactic acid production would be impossible. For case 4, epinephrine signals the fight or flight response within the human body. An increase in blood glucose is one of the body's responses to epinephrine. Therefore, an injection of epinephrine should stimulate the production of glucose through gluconeogenesis or glycogenolysis. Based on this, one would suspect that the patient is experiencing a disorder in which the process of converting glycogen to glucose is altered. For this reason, Lactosemia or fructose intolerance could not be a diagnosis despite the enlarged liver being present in both disorders. Lactosemia and fructose intolerance are both disorders in which the metabolism of certain sugars is affected due to a deficiency. Hereditary fructose intolerance is a disorder that is caused by the inability to convert fructose to glucose due to a deficiency in the allelase B enzyme. Galactosemia is caused by the inability to convert galactose to glucose, which is due to a deficiency of the galactose-1 phosphate uridyl transferase enzyme, also called the GALT enzyme, GALT. These two disorders have no effect on the conversion of glycogen to glucose. Furthermore, the proper test for the diagnosis of both of these disorders is a genetic test because they are both autosomal recessive traits. In addition, the diagnosis could not be determined by injecting epinephrine because epinephrine has no effect on the metabolism of fructose or galactose. Furthermore, with galactosemia, the patient would not experience an increase in blood glucose following breastfeeding because galactose is present in breast milk. The mom mentions that the patient is fine after consuming breast milk, but shortly after, the patient experiences symptoms of hypoglycemia. If the patient did have galactosemia, the patient would also experience hypoglycemia after breastfeeding because breast milk is a carb and galactose is an important sugar in breast milk, which further disqualifies it as being an answer. Glucose 6-phosphate dehydrogenase is used in glycolysis to produce energy in the form of ATP or to store energy in the form of glycogen. However, glucose 6-phosphate dehydrogenase is mostly known for preventing hemolytic anemia by inhibiting the buildup of reactive species within the cells. Glycogen 6-phosphate dehydrogenase may have an impact on blood glucose regulation in a fasted state in very extreme conditions. However, because the patient has no symptoms of hemolytic anemia, this disorder is very unlikely to be the correct diagnosis. Glycogen storage disease is a disorder in which glycogen is not properly stored within the body due to glycogen synthetase deficiency. It is important to note that blood glucose, as mentioned earlier, is maintained through glycogenolysis that occurs in the liver during the fasted state. The patient most likely has glycogen storage disease 1 and is deficient in glucose 6-phosphate enzyme. As a result, glycogen is unable to be broken down and released from the liver in order to maintain blood glucose levels. For this reason, there is no change in blood glucose once injected with epinephrine, and this inability to use glycogen causes an increase in lactic acid, which causes lactic acidosis. As a result of the buildup in lactic acid, the liver becomes enlarged. Glycogen storage disease is further shown to be the correct answer because, as mentioned earlier, administering epinephrine should cause an increase in blood glucose because epinephrine initiates glycogenolysis. So it should be expected that blood glucose increases after epinephrine is administered. But because it does not, it is unaffected. That is another example explaining